So today we're going to talk about lovastatin, the drug, and how it's effective in lowering cholesterol levels in the blood. So statins are a class of drugs that act on specific proteins in the cholesterol biosynthesis pathway. Doctors prescribe statins to patients who have high genetic cholesterol in hopes of lowering cholesterol in their blood. So this is specifically drugs are used for patients who overproduce or oversynthesize cholesterol and not those who overeat it. So statins do not lower high cholesterol levels for patients who have dietary high cholesterol. For these patients, doctors suggest a change in diet simply. But high cholesterol levels across the board increase risk of heart attack, stroke, and heart disease. And according to WebMD, statins are found to reduce the chances of death by these diseases by 25 to 35%, which is quite significant. And personally speaking, working in an ER, I see the med lists of a lot of our patients, and a lot of the patients, adult patients specifically, are on statins for their high cholesterol. So next we'll talk a little bit about cholesterol, which plays an important role in membrane structure, and it's a precursor to the synthesis of steroid hormones, bile acids, vitamin D, as well as other things, as we learned in our book and shown here. Less than 1/2 of cholesterol in our bodies is from biosynthesis de novo, which is, again, not dietary cholesterol, not cholesterol that we consume, just cholesterol that we synthesize in our body. So next, here's the full cholesterol biosynthesis pathway. We're going to focus on the initial steps. So the initial steps of the cholesterol biosynthesis pathway is called the mevalonate pathway, which we're going to talk about. But generally speaking, I can walk you briefly through this pathway. First, acetyl-CoAs are converted to HMG-CoA, or that's what we typically call it, but it's 3-hydroxy-3-methylglutaryl CoA. Then next, HMG-CoA is converted to mevalonate by way of HMG-CoA reductase. Next, mevalonate is converted to isopentenyl pyrophosphate, or we mostly refer to that as IPP. Then IPP molecules are converted to squalene, and squalene is finally converted to cholesterol. So that's a brief walkthrough of the cholesterol biosynthesis pathway. Statins act on specific proteins in this cholesterol biosynthesis pathway, specifically targeting HMG-CoA reductase. So this HMG-CoA reductase is the specific target of lovastatin. HMG-CoA reductase is required for cholesterol synthesis because it converts HMG-CoA to mevalonate. So those were in the early initial steps of the cholesterol biosynthesis pathway, which we called the mevalonate pathway. So this inhibits the conversion of the HMG-CoA to mevalonate, but it has a lot of effects down the road in the pathway, ultimately the inhibition of the biosynthesis of cholesterol. So lovastatin acts as a reversible competitive inhibitor for HMG-CoA. So the lovastatin binds to HMG-CoA reductase instead of the HMG-CoA. Lovastatin and HMG-CoA are structurally similar, shown here. So that's how lovastatin acts as a reversible competitive inhibitor for specifically HMG-CoA. So we'll move on next, but you can see there how the structures are very similar. So lovastatin is a prodrug. It's administered in its inactive form as an inactive lactone, or its native form, as we say. So in this form, up top, it has a gamma-lactone closed ring. After administration, though, the gamma-lactone ring is hydrolyzed in vivo, and it opens the ring, giving it a hydroxy acid, or it puts it into its active form. So that's what we see at the bottom. Here at the top it says that's lovastatin, then the ring opens and that's its new structure. It has a hydroxy acid, its active form. So this is hydroxy acid on the bottom is the species that actually binds to the protein and contributes to this inhibition of the synthesis of cholesterol. So in the end, the hydroxy acid, the active form of lovastatin, stops the reaction, acting as an HNG-CoA reductase inhibitor, which inhibits the production of mevalonate, then IPP, then squalene, and then eventually halts the biosynthesis of cholesterol. So that's a brief overview, but I hope you understand, and we'll see you next time.